I'd like to introduce our next presenter, Dr. Brian Pickles, and is an associate professor of ecology at the University of Reading in the UK. His diverse interests span plant-fungal symbiosis, forest dynamics, reptile conservation, and paleoecology. Brian?
Okay, can everybody hear me clearly? All good? Great. It's a real pleasure to be here. It's 7 years since my wife and I were last here in Texas doing some research with BCP, with Jim and Lisa. And in that time, we've had 2 kids, so if you hear some complaining at the back, that'll be my daughters back there. So we're very, very pleased to be here, and it's a real pleasure to talk to you. So a lot of the research that I do takes place below ground. I'm interested in below ground symbioses. Just to give you a little brief introduction to me, I'm an associate professor of ecology in England. You probably can't make it out. There's a dinosaur mummy hidden in this photo. That's one of the things that I work on these days. But a lot of my research interests are biodiversity, quite broadly, below ground biodiversity especially. I do a lot of work on plant-fungal symbioses and on paleoecology. And I'm fortunate to have some really brilliant PhD students working on diverse topics like graveyard biodiversity, reptile paleoecology in the Pacific Northwest, plant-fungal communication. We're trying to figure out how plants and fungi actually talk to each other. And I'm also really interested in how mycorrhizas can be used to help with woodland expansion. Because we're trying to make our woodlands bigger in the UK. They're all tiny little patches. So that's what I do, and my wife, Monica Gorzlak, is a research scientist with Agriculture and Agri-Food Canada. She works on soil microbial ecology, regenerative agriculture, soil health, so if you're interested in those topics, please have a chat with her. And she's working on things like mapping soil carbon, pesticide degradation, and looking at mycorrhizal networks in intercropping, and that's her team there. So again, we were fortunate to come out to this part of the world 7 years ago and work in these really interesting juniper-oak woodlands. So juniper and oak co-occur throughout North America, as I'm sure you're aware. This is the distribution of juniper, and so you've got various different phylogenetic groupings here. And here is a map of the location and number of oak species in the Americas. And what I think is really interesting about this part of the world is you've actually got really high species diversity of oaks in this area, and you've got really high phylogenetic diversity of junipers. So they're occurring in these woodlands throughout this area. Okay, a little bit of audience participation. What's this? A tree? I would argue that's half a tree. And this is the bit that nobody ever thinks about, okay? And this is what I work on, but really, that's not even half a tree, or that's not even 100% of a tree, because you've also got all of this going on below ground. Lots of important fungi in the soil that are associating with different trees, helping them to get nutrients out of the soil. So when you are looking at these wonderful ash juniper and your wonderful oaks in these ecosystems, don't forget you're only seeing half the story. There's a lot going on below ground. In fact, if you go down into some of the limestone caves, you'll find roots of big oak and juniper popping down in there. So if you take nothing else away from this talk, remember that there's a lot going on below ground that normally you just don't see. Okay, no, I'll keep going. Soils. There's been some great talks already about soils in the Edwards Plateau and the range of different soils that you see, from really nice rich organic soils to very poor low carbon soils. Now, this is a photo from about 7 years ago. I think Daryl might have gotten over the work that we were doing here. Here we're digging up individual seedlings. This is a 4-year-old oak seedling. And it was already about 70 centimeters down through multiple limestone layers, requiring hours and hours. It took us 12 hours to get some of these seedlings out after just 4 years. So they're making their way down through the limestone. So of course, soils combine biology, chemistry, and physics. They provide habitat and nutrient sources for a lot of other organisms. They're the substrate on which the plant communities are growing. And they can also provide us with a record of the past. So you can collect climate proxies, you can collect vegetation community proxies and things like this from soils. Fossils, one of the other things that I work on. You need to deal with soils if you're going to deal with these different things. And soil fungi are particularly important in forests. They provide a whole range of different positive, neutral, and negative services. So we've got mycorrhizal mutualisms. These are typically positive for trees. We've got saprotrophic fungi that are actively breaking down dead biomass. And these can be neutral to positive. As they break down biomass, they're putting nutrients back into the soil. And we also get pathogens, of course, which can cause the die-off of certain species, which can then benefit other species. But if you're the thing that's being attacked by a pathogen, generally that's bad. Okay, so soils. Roots and mycorrhizas. It's mainly mycorrhizas that I'm interested in. Oftentimes when people think about plants, if they ever think about the roots of the plants, it's sort of assumed that they're doing all of the work of picking up nutrients and all that sort of thing. Well, roots are providing stability and they're exploring the below-ground environment. But for most plant species, certainly for most trees, they're not using roots to collect nutrients from the soil. They are associating with fungi and getting the fungi to do the hard work of collecting nutrients for them. So here we've got our tree with lots of fungi associated with it. There are sort of two main types of these mycorrhizas. Mycorrhiza means, it comes from the Greek, fungus root, and it consists of a mycorrhizal fungus and a plant root. And I spent my PhD looking at many of these, 150,000 root tips I looked at. These are a few images. They kind of look a little bit like corals on the tips of roots. And you can actually, most of the time, see them with the naked eye. So ectomycorrhizas, they're usually scaled to a few millimeters. The other major type of mycorrhizal association that you find is arbuscular mycorrhizas. This is an image of an arbuscule inside the cell of a root. So ectomycorrhizas and arbuscular mycorrhizas. And the important thing to bear in mind in the context of these Juniper-oak woodlands is that oaks associate with ectomycorrhizas and junipers associate with arbuscular mycorrhizas, or that's the story anyway. So mycorrhizal fungi, they associate with most plant species. Arbuscular mycorrhizas associate with an overwhelming majority of plants, ectomycorrhizas with very few, but the species that ectomycorrhizas do associate with tend to be the large dominant woody species. They exchange nutrients with the plants. So when a plant photosynthesizes, it produces lots of nice sugars that it gives to the fungi in exchange for nutrients, sometimes water, protection from pathogens, and there's various different signaling molecules that get transferred around as well. They are patchily distributed in the soil. So this is an image of a 20 meter by 20 meter patch of a woodland. You've got high density of fine roots, you've got low density of fine roots, and layered on top of the distribution of the fine roots are the different species of fungi that are competing with each other or facilitating each other. Again, as you go further down through the soil, you get different communities of fungi at different depths doing different things. So, complex variable environment that you can't just look at. And of course, it gets even more complicated when you start to consider that you've got fungal mycelium colonizing roots of a plant, and a single fungus can connect multiple roots within one plant. And sometimes they can connect a single mycelium between multiple individual plants. And then you've got multiple species of them per plant. Some of them associate with different types of mycorrhizas as well. So very quickly, you get lots and lots of interactions going on. And then, of course, we have more complex ecosystems. We've got our trees, we've got our seedlings and shrubs, we've got grasses, we've got all sorts of different layers of these fungi associating with each other and with their plant hosts below ground. So you end up with a situation where you've got roots of multiple species of plants connected by fungi. Things like orchids, for example, which were mentioned briefly in a previous talk. Most orchids steal carbon from fungi as part of their life cycle. This is called mycoheterotrophy. Some plants entirely rely on carbon that they steal from fungi. And those fungi are connected to trees, they're getting their carbon from the trees, and then the orchids are tapping into those fungi and getting their carbon from there. This is a really dynamic environment. It's a dynamic environment, it's below ground. You've got fungivores, you've got roots turning over, you've got pathogens, you've got antibiosis. So these networks and these fungi are always in flux. And remember that fungi are really good at picking things up from the soil. So they're acting kind of like sponges. Okay, so 7 years ago when we were doing some research in the BCP, we were looking at fungal interactions in these oak and juniper woodlands. And what we were finding, one of the key findings, is that when we have more mycorrhizas, we see fewer pathogens in the soil. So having mycorrhizas is really good for helping to protect you from pathogens. And we would see a really high abundance of ectomycorrhizal fungi and also saprotrophic fungi under living canopy, which makes a lot of sense. We also find a high diversity of saprotrophs when you have a dead canopy. Again, they're breaking down coarse woody debris. They're converting that biomass into nutrients. So if you start getting rid of your dead trees and your coarse woody debris, then the saprotrophs can't get in and unlock those nutrients. And we also found that pathogen diversity increased the further down into the soil you went. And you would also find where canopies were dying off or where you have lots of grasses, we were finding more pathogens associated with the roots of the plants. We also looked at some of the fungal communities associated with these mature roots in caves, and this is where it got really interesting. So you get the roots of these mature oak and mature juniper popping down into limestone caves. You can see there's different soils that are associated with them. There's lots and lots of saprotrophs in these caves, as you might expect. What we didn't expect was the diversity of ectomycorrhizal fungi that we were finding associated with the roots and soils in these caves. Sometimes you'll even get sporocarps of ectomycorrhizal species that will actually grow within a cave. And the really cool thing was that we found some species that were associated with oak, some species that were only associated with juniper, and some species that were associated with both oak and juniper. And these are all ectomycorrhizal species, and if you remember from earlier, juniper doesn't associate with ectomycorrhizal species. We also found the arbuscular mycorrhizas that we were expecting to see associated with the juniper. So what's going on here? Well, one of my favorite people who I work with, Professor Mark Tibbett, he likes to say, the problem with fungi is that they haven't read any of our textbooks. So, we see various different types of facilitative effect between ash juniper and plants. When the canopy is removed, juniper is often the first to colonize, like you see on some of the balconies here. And some of the work that we've done shows that oak seedling survival and ectomycorrhizal colonization of those oak seedlings increases as you have more ash juniper in the surrounding area. Also shrubs, also living oak, but the ash juniper appears to be an important part of the facilitation. And we published some work on this in 2020. To me, the really curious thing is that we're seeing ectomycorrhizal fungi associated with juniper. Now, that doesn't mean that they are forming ectomycorrhizal associations. We're not entirely sure what they're doing, but they are interacting in some way. And of course, one of the other things that we're seeing is that when you have more mycorrhizas, you have fewer pathogens. So potentially that's a part of a facilitative effect that's going on. And it's not just here that we are seeing these interactions. There's a paper on Mexican Juniper-oak woodlands supporting evidence of ectomycorrhizas interacting with alligator juniper in these Mexican juniper-oak woodlands. And we're seeing potential interactions between juniper and oak in the Balcones Canyonlands, in these Texan juniper-oak woodlands. And in fact, if you go back in the literature, there's some early work on Juniper Phoenicia in Europe, which suggests an ECM, an ectomycorrhizal interaction, but subsequent studies rubbished that because, well, juniper's not supposed to associate with ectomycorrhizal fungi. So whether that's true or not, we're not entirely sure. So the take-home message is, soils are really important, but they're often forgotten. It's easy to forget about things that are really hard to study. Who wants to spend 12 hours of a day digging up seedlings other than me and Daryl and Monica and a few other people? But soil organisms play these really essential roles in ecosystems, and we often completely ignore them. And those roles can be positive, they can be neutral, they can be negative. Mycorrhizal fungi are really important. They support our oaks, they support our junipers, they support rare species like orchids. And maybe there's some unexpected things that they're doing in these ecosystems that haven't been well described because people haven't been looking at them and they've been told that they shouldn't look for them. So maybe that's one of the reasons for some of these facilitative effects that we're seeing. That's all the time that I've got. Thank you very much for listening. Thanks to our collaborators and funders. Okay, great. Any questions? There's one over here. I'm going to say it would be very speculative to say that there's a clear association there. But knowing what we know about all of these complex interactions, it's entirely possible that, you cut down ash juniper because you want to encourage the growth of oak, and, well, you've just lost some other member of the community that was below ground that you didn't even know was important. So possibly, but I'm not aware of any research that's been done on that. If only there were a way to just see the ectomycorrhizal fungi. Many species produce above ground sporocarps, so your mushrooms, so you can do fruit body surveys. I mean, you had some rain here recently, and just going out walking around, you will find mushrooms, you will see sporocarps. Some of those will belong to ectomycorrhizal species. If you want to see an ectomycorrhizal interaction, then you need to dig down and have a look at the roots. And if you want to figure out what they're actually doing, what the host and the fungus is doing, that's a whole other realm of complicated, different scientific approaches. In terms of ease of seeing them, fruit body surveys and just dig in the soil until you see them. That's a great question, and that is what one of my PhD students is doing back in the UK, is looking at the idea of using mycorrhizas for mediation, specifically for woodland development. It's interesting because most of these ectomycorrhizas that I've been talking about have evolved from saprotrophic fungi. So at some point they have switched from degrading wood by themselves to collecting other nutrients that have been broken down by other saprotrophs, although some of them retain the ability to break down wood themselves. In terms of actually using them as amendments, that's something I'm working on at the moment. So we're collecting sporocarps from a local area and even using soil amendments, and then seeing if we inoculate seedlings with those different either mixes of mushrooms or with soil that we've collected from under our target tree species, do we actually see an improvement in the survival or the growth of our seedlings? So from my perspective, that works at an early stage, but there are quite a few people who are looking at these sorts of impacts in different ecosystems. Good. Thanks.